Hello and welcome to Sam Biology class. Friends, today we shall discuss about beta oxidation of fatty acids. Fatty acids are oxidized by most of the tissues in our body. However, brain, erythrocytes and adrenal medulla cannot oxidize fatty acids for energy requirement. Fatty acids are mostly oxidized by beta oxidation pathway in which fatty acids are oxidized on beta carbon atom. Each cycle of beta oxidation results in the sequential removal of a 2-carbon fragment acetyl coenzyme A. The process of beta oxidation involves 3 stages. In stage 1, fatty acids are activated to acyl coenzyme A. This occurs in the cytosol. In stage 2, acyl coenzyme A, which is the activated form of fatty acid, is transported from cytosol into mitochondria. Stage 3 is beta oxidation proper, which occurs in the mitochondrial matrix. So stage 1 is activation, which occurs in the cytosol. This takes place in 2 steps. Both the steps are catalyzed by the enzyme thiokinase. In first step, fatty acid react with ATP to form acyl adenylate. In this step, ATP is converted to inorganic pyrophosphate, which is then hydrolyzed by the enzyme pyrophosphatase to form 2 inorganic phosphate, thereby making this activation process totally reversible. In second step, acyl adenylate react with coenzyme A to form acyl coenzyme A. So, acyl coenzyme A is the activated fatty acid. Next, stage 2. Inner mitochondrial membrane is impermeable to fatty acids. Because of this, the activated fatty acids, that is the acyl coenzyme A, is transported into the mitochondria by a specific carrier system called carnitine shuttle. This occurs in 4 steps. In first step, acyl coenzyme A react with carnitine to form acyl carnitine. This reaction is catalyzed by the enzyme carnitine acyltransferase 1, which is present on the outer surface of the inner mitochondrial membrane. In second step, acyl carnitine is transported into the mitochondrial matrix by a carrier protein. In the mitochondrial matrix, acyl carnitine reacts with coenzyme A to form acyl coenzyme A and carnitine. This reaction is catalyzed by the enzyme carnitine acyltransferase 2, which is present on the inner surface of the inner mitochondrial membrane. In fourth step, carnitine is transported back into the cytosol to continue the process. After transport into the mitochondrial matrix, acyl coenzyme A is oxidized by beta oxidation proper, which consists of a sequence of 4 reaction steps. In first step, acyl coenzyme A is oxidized to delta 2 transaenoyl coenzyme A. This reaction is catalyzed by acyl coenzyme A dehydrogenase, which is FAD dependent. In this step, FAD is reduced to FADH2. Second step is the hydration of delta 2 transaenoyl coenzyme A to beta hydroxyacyl coenzyme A. This reaction is catalyzed by the enzyme enoyl coenzyme A hydratase. In third step, beta hydroxyacyl coenzyme A is dehydrogenated by the enzyme beta hydroxyacyl coenzyme A dehydrogenase to beta ketoacyl coenzyme A. This enzyme is NAD dependent. In this step, NAD is reduced to NADH. Finally, in the fourth step, beta ketoacyl coenzyme A is cleaved by the enzyme thiolase, yielding an acetyl coenzyme A and an acyl coenzyme A, which is shortened by 2 carbon atoms. These 4 reaction steps constitute a cycle of beta oxidation. So each cycle results in the sequential removal of a 2 carbon fragment, acetyl coenzyme A. The scheme of oxidation is applicable to the even carbon saturated fatty acids. An example is palmitate, which is a 16 carbon saturated fatty acid. After its activation and transport into the mitochondrial matrix, palmitoyl coenzyme A undergoes 7 cycles of beta oxidation yielding 8 acetyl coenzyme A, 7 FADH2 and 7 NADH. So friends, this is about beta oxidation of fatty acids. For more updates, please subscribe some biology class.